Today we'll be going over exercise 15, mitosis. It does start on page 153 and we'll be following along pretty well in the book. You'll definitely want to read over exercise 15, but if you need some more help, you might check your lecture instructor's notes. There's also the Amoeba Sisters mitosis video. Good luck typing in the tiny URL. Khan Academy also has a more in-depth series of videos on cell division. If you're having any trouble, check out those two videos. You will need to know the phases of mitosis listed in table 15.2 on page 155. We've also got some pictures of some models, and while we'd normally have some slides, you might want to take a look at some slides in the science tutorial lab. We've at least got pictures of slides today. There are some definitions you should probably know about before we get started. Mitosis really is the division of the chromosomes which are in the nucleus. Cytokinesis would be the division of the cytoplasm and all the other organelles. At this point, we're just going to say magic happens. I bet there's a big long process, but right now, magic happens. We will be talking a lot about things that are happening in the nucleus. The nucleus does contain the DNA. We're going to be dividing the DNA or chromosomes out, so we'll be talking about the nucleus. Chromatin is a diffuse mass of DNA. Ms. Crotty compares it to a bag of cotton candy. Nice, fluffy cotton candy. A chromosome is going to be condensed DNA. Ms. Crotty says that's when you give the bag of cotton candy to your 5-year-old who wads it up into a little ball. That would be condensed cotton candy. It's still the same cotton candy, but it's not near as fluffy. We will talk a lot about homologous chromosomes. That's going to be a pair of similar chromosomes. Us humans are diploid. We have 2 copies of every chromosome. You get a copy of chromosome 1 from mom, you get a copy of chromosome 1 from dad. That would be a pair of homologous similar chromosomes. If the gene for hair color is on chromosome 1 in mom, it's also on chromosome 1 in dad. So they've got similar chromosomes. Now they may not be exactly precisely the same. When we start talking about genetics, we'll talk about different alleles and how maybe mom has brown hair and dad has black hair. There would be similar chromosomes with the same sorts of genes, but not necessarily identical genes. Sister chromatids, however, would be some identical chromosomes. One of the first things we'll have to do is, to make 2 cells, we'll need to replicate some DNA. Yes, the A's still pair with T's and G's still pair with C's, but we'll be making identical chromosomes. If mom's chromosome 1 has the allele for brown hair on it, then mom's sister chromatid will have the same identical genetics, i.e. the brown hair allele. If dad's chromosome 1 had the allele for black hair, then dad's sister chromatid will also have the allele for black hair. What? Wait, shouldn't it be dad's brother chromatid? Sorry, they call them all sister chromatids, whether you're getting them from mom or dad. The nucleolus, you might remember, is the site of RNA manufacture in the nucleus. We'll see that coming and going, so yeah, just wanted to remind you of that one. We'll also be talking about centrioles and spindle fibers, which pull chromosomes apart. You might remember from exercise 5 that centrioles function in cell reproduction. Welcome to cell reproduction. We'll be using the centrioles and spindle fibers to pull chromosomes apart. At the end of mitosis, a cleavage furrow will form where the cell pinches inward to form 2 cells. That only happens in animal cells, while the cell plate forms from the middle outward to divide into 2 cells in plant cells. We'll talk about that eventually. Table 15.2 on page 155 does have the phases of mitosis. We'll be talking about the phases of mitosis in animals first, and then we'll come back around to plant cells. During interphase, first of all, the nucleus is distinct. On the picture, you can kind of see the little gray ball where somebody drew a nuclear membrane with a pencil. I threatened to, it wasn't me. Chromatin is present. See these slightly medium blue stringy things that are in there? No, they're not real easy to see. That's the chromatin that's present. Eventually, when the cell gets ready to divide, it will replicate its chromosomes, and there'll be 4 copies of every chromosome, 2 from mom and 2 from dad. Those would be usually the chromatids, but they are replicating. The nucleoli is visible. See, there's a little red dot in the center? That would be the nucleolus, the site of RNA manufacture. Because the cell is doing normal things, it's producing all its proteins, it's making all its RNA and producing proteins, and everything's working just fine. Centrioles are visible next to the nucleus. There's some little black dots just to the left of the nucleus there. Those would be the pair of centrioles that will function in cell reproduction. For the most part during interphase, the cell is doing normal things. If it's a skin cell, it's being a skin cell, protecting your body, keeping things inside the body, keeping things outside of the body. If you're a bone cell, you're protecting. If you're a muscle cell, you're moving, but you're doing normal cell things. The big thing that happens during prophase is the chromatin, the diffuse mass of DNA, coils to form visible rod-shaped chromosomes, the condensed DNA. This is where your 5-year-old is wadding up the cotton candy, so that you can see the individual chromosomes. On this slide, you can make out the gray-blue chromosomes, and you can actually see where they're at this time, rather than just being a pot of cooked spaghetti noodles. You can actually see individual noodles in there. The nucleoli, the red dot, has disappeared. If you're going to do cell division, you should probably stop making some RNA and stop making some proteins and start dividing the cells and the chromosomes. The third thing is the nuclear membrane has disappeared. We did not draw a pencil mark around there, so it's kind of gone. So there you go, the nuclear membrane dissolves. Centrioles with asters will divide and migrate to opposite sides of the pole. Our little black dots are now at 12 and 6 o'clock, north and south. Little dots on the top and the bottom. They're going to be pulling on the chromosomes. But the big thing in prophase is that you condense into chromosomes. During metaphase, spindle fibers radiate out from the centrioles and pull on the chromosomes. That's kind of the pink blob in the middle. They're kind of playing a tug-of-war between the centrioles and the chromosomes, and as long as there's equal pressure from both sides, the chromosomes do line up nicely in the middle. Okay, the lab book says the chromosomes align on the equatorial plate, equator, middle of the earth. I'm going to use middle so that we can remember middle. M is for middle and M is for metaphase. Spindle fibers are also attached to the centromeres. We weren't really going to talk about the centromeres, but yes, there is a particular spot on the chromosome that the spindle fibers attach to and pull from. My poor model here has fallen a time or two on the lab bench, and he's missing a few parts of his chromosomes, but you can still see the blue-gray chromosomes across the middle of the cell. During anaphase, the big thing is the daughter chromosomes move apart by the aid of spindle fibers to the opposite poles of the cell. A is for anaphase, A is for apart. On the picture here, you can kind of see a little bit of pink in between those blue-gray chromosomes. If you see any kind of space between the chromosomes, we're going to go ahead and call that anaphase. But basically, those centrioles and spindle fibers are doing their jobs and pulling the chromosomes apart to the top and to the bottom. Yes, the centromeres split and separate, and science majors talk about exactly how many chromosomes you've got in any given phase. In telophase, or telophase, fine, whatever. In telophase, the daughter chromosomes reach the opposite poles of the cell. You can see we're fixing to become two cells here. The chromosomes are really far apart. They will uncoil any minute now. The nuclear membrane will start reforming. The nucleolus will come back, and pretty soon you'll have two cells. Note you've kind of got some pinching inward on the left and the right side. That's the cleavage furrow, the pinching inward that's going to form the two cells. That happens only in animal cells. Once telophase is done, you do have two identical daughter cells. They are identical cells that are half the size of the original cell. They've got an identical number of chromosomes, and the chromosomes are the same. If you had a copy of mom's chromosome 1 with the brown allele and dad's chromosome 1 with the black hair allele, you'd have the same chromosomes in each of the two cells. They are identical to the original cell. In real life, they're about half the size. In plastic model life, ooh, these guys are probably about the size of the mother cell. Sorry about that. They have not quite got their nucleolus back in this picture, and they haven't quite got their nuclear envelope back or nuclear membrane back, but you can already see the centrioles are on the sides there. So they're pretty close to being fully fledged daughter cells that just happen to be in interphase beside each other. The following cells represent plant cells. Note the rectangular shape on these, whereas we had the round shape on the cells before. Interphase in plants is going to be fairly similar. There's still a nucleus that's distinct in the middle. The chromatin is present, although it's in black chromosomes, or chromatin this time. The chromosomes will replicate, so there'd be 2 copies of everything. The nucleoli are visible. This one's more of an orange blob than a little red dot, but the nucleoli are visible. Wait a minute. There were 5 things on interphase in animals, and there's only 4 things in plants. There's no centrioles. What's up with that? Aren't the centrioles the things that pull the chromosomes apart? Yes, there has got to be something in there. It is not officially a centriole, but there has got to be something in there. I've been betting for the last 10 years that they would find something any minute now and put a name on it, but still haven't heard anything. I'm convinced there's got to be another organelle in there somewhere, and scientists just haven't quite found it yet. They'll probably have to come up with a new name for it in plants too, because it is not a centriole, but it is something that functions like a centriole, is my guess anyway. Here's a prophase model for plants. The chromosomes are starting to condense so that you can actually see them. And really you can see they come in pairs. There's the set of 2 black lines there. That would be the duplicated chromosomes. The nucleoli, the orange dot, is starting to disappear, or at least there's a chromosome sitting on top of it. The nuclear membrane in red around the nucleus is mostly gone. You can see the spindle fibers are starting to form and pulling towards something on the left and right side of that cell. Maybe it's not a centriole, but there has got to be something in there. During metaphase in plants, like the animal cells, the chromosomes are going to line up in the middle. The spindle fibers go to something that's not a centriole, and the centromere, and do the tug of war, and the chromosomes do end up lining up nicely in the middle, on the equatorial plate. Fine, whatever. They'll line up in the middle. During anaphase of plants, we'll be pulling the chromosomes apart, just like we did in anaphase with animals. As long as you can see a little bit of space between the black chromosomes, we're going to call it anaphase. The centromeres split and separate, the daughter chromosomes move by the aid of spindle fibers to opposite poles of the cell, and you have a little bit of space as the chromosomes are pulled apart. A is for anaphase, A is for apart. During telophase, the daughter chromosomes reach the opposite poles. The chromosomes are starting to uncoil and go back to chromatin. The nuclear membrane has not formed on this picture. Imagine a red line around the 2 nuclei soon. The nucleoli have not become visible just yet. The other thing that's happening is that silver line down the middle. That's the cell plate that forms from the inside and spreads towards the edges. I still find it interesting that the animal cells will pinch inward until you get 2 cells, whereas the plant cells will kind of grow from the inside out. They're kind of opposite that way, but it works. Like the daughter cells of animal cells, the daughter cells on plants are identical cells that are about half the size of the mother cell. They've still got an identical number of chromosomes as the original cell had, and each individual chromosome is identical to what the mother's chromosome had. Notice these really are 2 cells. We've got the red nuclear membrane around both of them. We've got the chromatin back. We've got the nucleoli back. And we do have a complete cell wall between the 2. Well, they sure look like squares now instead of rectangles. They'll get bigger and become rectangles. but we do have daughter cells that are identical to the original cells. Be sure to look over pages 154 to 157 with figures 15.1 through 15.6. It's got all the different phases in plants and animals. Oh, there's interphase, there's prophase with a middle and late stage for some reason, there's metaphase, there's anaphase, there's telophase, there's daughter cells. Look over the pictures and see if you can identify the parts. We might put a picture of one of the figures on the lab exam, and you should be able to identify the phase of mitosis from said pictures. In a real Biology 1408 lab, we'd have made you look at the animal mitosis slides. I always had some pictures, so here you go. You're also welcome to go to the Science Tutorial Lab, SSCI, room 1501, and look at some slides on your own. We do have the animal mitosis slides here. They're kind of hard to see sometimes. One of the cells in the middle is labeled with prophase. You can see it's a kind of round cell, and it does have a darker round blob in the middle. Yeah, if all you can see is blob in the middle, that's probably prophase. The cell just to the right of it is going to be metaphase. It's not real easy to see, but the chromosomes do line up from about, oh, I don't know, 8:30 to 2:30? 9 to 6? Left to right? The chromosomes are lining up in the middle there. On the upper left-hand corner is a nice anaphase. The chromosomes are pulling apart. No, there's not a heck of a lot of space between the two sets of chromosomes, but there is a little bit more space between those chromosomes than there's no space on that metaphase one. So anaphase is pulling apart. Here's a better view of an animal cells in anaphase. Those two sets of chromosomes are definitely pulling apart, and there's no real pinching inward or anything, so we've got a really nice, pretty anaphase at the black arrow. Here's a, well, not so great telophase. The cells are moving apart from, I don't know, about 11 o'clock to 5 o'clock. The chromosomes aren't lining up real well so that you can see them, but there's some pinching inward in the kind of axis on the, I don't know, 2 p.m. to 7 p.m. line. You do get some pinching inward, and you can actually see a pretty good cleavage furrow on these two cells. Well, one cell that's fixing to become two cells. We're pulling really far apart and just about to be two cells. The highlighted cell is a plant cell in prophase. They are kind of rectangular cells there, so it is plant cells. And like I said, if you see a blob in the center, that's probably just going to be prophase. Honestly, most of the cells on this slide are probably prophase as well. Maybe a couple of interphases, but most of them I would call prophase. I stole this slide off the internet. Somebody had a good microscope with some really nice resolution. But you can see the chromosomes are lined up fairly well in the middle. Sometimes the plant chromosomes are a little long and they don't line up exactly precisely in the middle. I don't know, if you asked kindergartners to line up in the middle of the room, I bet they form a really nice line. If you asked college students to line up in the middle of the room, somebody's wibbling, somebody's wobbling, and the line's just going everywhere. I think that one's college students lining up in the middle. But it's as close as it gets to the chromosomes lining up in the middle, so we are going to call it metaphase. Here's a really nice plant cell in anaphase. Note the chromosomes are pulling really far apart. If you're thinking you might see something in the middle like a cell plate, that's a figment of your imagination. Sorry, we're going to call this one anaphase. Yes, that cell plate is going to form any second now, because the chromosomes are really far apart. But we've got a nice anaphase here. Here's a plant cell in a really late stage of telophase. You can see the two nuclei there, and there is a line almost all the way across in the center. It's well on its way to becoming two cells, awfully close to daughter cells. I don't know, maybe another 20 minutes or so, and you'll actually have two daughter cells there. But if you do see the chromosomes really far apart and starting to unwind back into chromatin, and especially that cell plate in the middle, we're going to call that telophase. On a lab quiz or a lab exam, we could ask a bunch of questions about these things. We could start with, is this a plant or an animal cell? It's an animal cell. It's round. Using the same slide, we could ask what phase of mitosis is the cell at the arrow. It is lining up in the middle. That middle kind of varies as to whether it's up and down or left and right, but that would be metaphase. What about the cell to the right of the labeled cell? That's another metaphase. How about the cell to the left and underneath? That one's a little harder. It's a blurry picture. It's not the greatest picture, but it is anaphase. The chromosomes are starting to pull apart kind of top and bottom. Any of the rest of the cells, I'm going to say, are just in interphase. You can't even see the blob for the nucleus for prophase. More questions. What phase of mitosis is the cell at the blue arrow? It's pulling apart. Apart is anaphase. What about the red arrow? The red arrow is kind of hard to see, but that one's probably going to be a telophase. He doesn't have his cell plate showing real well, so I don't really like the picture, but he's probably most likely in telophase. There's a green arrow. That one's probably metaphase, lining up nicely in the middle. On the bottom right-hand side, there is a black arrow that kind of blends in with all the rest of the cells. What phase of mitosis is that one? That's probably prophase, because you can see the blob. Will the red arrow cell form a cell plate or a cleavage furrow? We said it was telophase, but is it a plant or an animal cell? It's a plant cell. Plant cells are the ones that form a cell plate. On page 158, there's a big table where you can draw pictures of the phases of mitosis in plants and animal cells, so if you need to write any notes, that's a good place to do it. On page 159, there are 5 questions. You should be able to answer the questions, and yes, we copy some of these straight out of this lab exercise and put them on the lab quiz and the lab exam. So be sure to read the chapter, find the answers, and check with your instructor if you need any answers confirmed. Thank you.